Mitosis and meiosis, a further look into the cellular processes, their importance, and a comparison between the two. The cell cycle is the life cycle of the cell. It begins with interphase, which is separated into three parts, G1, S, and G2. In G1 phase, or the first gap phase, the cell grows and prepares for S phase. In S phase, or synthesis, the chromosomes are duplicated and the cell prepares for G2. In the second gap phase, G2, organelles are duplicated. The cell undergoes another stage of growth as it prepares for mitosis. If the cell does not pass through the checkpoints of interphase or is not to divide, the cell goes into G0. Following interphase is mitosis, which is divided into prophase, metaphase, anaphase, and telophase. After proceeding through this portion of the cell cycle, the parent cell divides into two identical daughter cells by cytokinesis, or the division of the cytoplasm. A cleavage furrow forms and the parent cell is pinched in half.
Mitosis is the cell division which results in two genetically identical cells. Mitosis is important because it allows for genetic stability, growth, and repair in an organism. The first stage of mitosis is prophase. The nuclear envelope disappears, the DNA condenses, and the chromosomes become visible. Mitotic spindles appear as the cell begins to enter metaphase. In this cell, there are 4 double chromosomes connected by a centromere. In metaphase, the chromosomes align along the metaphase plate. Each sister chromatid is attached to the mitotic spindle by a kinetochore. In anaphase, the proteins of the centromere linking the chromatids break apart and the sister chromatids separate, as meiotic spindles shorten. Anaphase is the shortest stage of mitosis. In this cell, there are now 8 chromosomes, or 2N. In telophase, the mitotic spindle disappears, the nuclear envelope reforms, and the DNA decondenses. Cytokinesis begins to occur with the formation of a cleavage furrow. Cytokinesis is complete with the division and separation of the cell outside the nucleus. Each cell has 4 chromosomes and is identical to the original cell.
Meiosis is the cell division resulting in 4 genetically diverse haploid cells. Meiosis provides genetic diversity, which promotes evolution. It is split into 2 stages, meiosis I and meiosis II. Meiosis I is the separation of homologous chromosomes. During this phase of meiosis, the cells are considered to be diploid, or 2N in chromosome count. Meiosis II, however, is the separation of sister chromatids. During this phase of meiosis, the cells are considered to be haploid, or N in chromosome count. During interphase, organelles and chromosomes are duplicated and the cell prepares for meiosis I. In prophase I, chromosomes condense and become visible. The nuclear membrane and nucleolus disappear and a spindle apparatus begins to form. Homologous chromosomes, or tetrads, align during a process called synapsis. Crossing over occurs. Genetic material is exchanged by the homologous chromosomes. There are 4 chromosomes in this cell, making it 2N, or diploid. During metaphase I, the tetrads move along their microtubule attachments, so they are lined up along the metaphase plate. The cell is still 2N. In anaphase I, the tetrads separate and are pulled to opposite poles of the cell. In telophase I and cytokinesis, the cells split apart, forming 2 daughter cells. The chromosomes uncoil, and the nucleolus forms, preparing for meiosis II.
Meiosis II separates sister chromatids. The first stage of meiosis II is prophase II. The nuclear envelope breaks down, and the chromosomes attach to the reforming spindles. Each cell has 2 double chromosomes. During metaphase II, the chromosomes align along the metaphase plate. In anaphase II, sister chromatids move towards opposite poles of the spindle. The sister chromatids are individual chromosomes when they separate. In telophase II, the chromosomes begin to uncoil and the nuclear envelope begins to form. Each cell has 2 chromosomes, half of what we started with, making them N. Cytokinesis occurs, resulting in 4 genetically diverse haploid cells.
Mitosis and meiosis. What do they have in common?
Mitosis and meiosis both occur on a cellular level and create new cells by separating old ones. Parent cells share genetic material with daughter cells, and both are required for life. Mitosis is a process of asexual reproduction that allows for the creation of identical cells in an organism. Meiosis is a process of sexual reproduction that allows for genetic variation in an organism, thus promoting evolution.